Welcome all to this special seminar here on a Thursday at 4 o'clock. I think the first announcement, first of all, I would like to thank Don and Steve for agreeing to come and give this seminar. We've been having scientific meetings all day, and they flew in yesterday lunchtime, and so it's very nice that they've agreed to actually give a seminar to all of us here, because they're flying out again, at the end of tomorrow. But before we start, I would just like to make the announcements. I was really keen that you all have an opportunity to meet Don and Steve. So the ASC Center of Excellence has set up some drinks in the tea room area, and I hope you do all take the time to join us for some drinks after the seminar series. Now, well, I can see all of you here, so it's really clear that neither Steve Long or Don Ort need a lot of introduction, and you've had this email which gave you a lot of background. Steve Long and Don Ort both placed at the University of Illinois in the USA. Steve also at Lancaster University, and they're famous for their photosynthetic research. But what always strikes me as one of the greatest achievements that I think they've managed to take our lab-level photosynthesis research out into the field, and they've demonstrated that enhancing photosynthesis or manipulating photosynthesis does actually have benefits when you go into the field. And I don't want to waste their time anymore, and I think we are starting with Steve.
Yeah.
I'll let you. Yeah.
Okay, thanks. I just thought I would start with this picture. This is where Don and I work, the Institute for Genomic Biology at the University of Illinois. But right in front here are the Morrow plots. That's actually the oldest agricultural experiment, except for the Broadbalk in Rothamsted. So ever since 1876, maize has been grown here continuously under a wide range of treatments. So at the university, we're very connected to the land, which is not surprising because Illinois is the largest producer of soybean in the United States and the second largest producer of maize as well. So about over half of our state in the summer is covered with those two crops, and 85% of it is transgenic. So if we put transgenic plants in the field, nobody in the local area has any problems with that, which is rather nice. We also have the National Center for Supercomputer Applications on our campus, which gives us high-performance computing, which has been very important in our work as well. So we're dealing with a problem which has varied predictions, but UNFAO have said by 2050 we're going to need to produce 60% more food than we are today, and we're not going to have more land to do it on, and we're also going to have to deal with climate change as well at the same time, and possibly diminishing water resources. Now, of course, we faced problems like this, or the world did, in the 50s, when there were serious famines in China, India, and so on. And the Green Revolution really took care of that. And so for a number of years, we had year-on-year increases in yield. And this just illustrates the situation for rice. This is decadal improvement in yield in the three largest producers of rice, China, India, and Indonesia. You can see in the 70s and 80s, decadal improvement was round about, averaging about 30%. But as we move into the 21st century, the first decade, these numbers are now diminishing to single figures. And maybe what is surprising is that China was investing more than 100 times what it was investing then to improve a yield. So it's telling us it's getting harder and harder to get improvements. Now, one simple way of looking at this is, what determines the yield of a genotype at a given location? If it has sufficient water, nutrients, it's free of pests and diseases. So this was an equation developed some time ago by John Monteith, where he said, well, the yield is, in that situation, the yield is determined by the amount of solar energy available during the growing season of that crop, multiplied by the efficiency with which the crop captures that radiation, multiplied by the efficiency with which it converts that radiation into plant biomass, multiplied by the efficiency with which it partitions that into the parts of the plant we care about, i.e. the grain of rice, the seed of soybean, and so on. So these are some numbers for a modern soybean crop growing on our university farm at Illinois. And you can see that if we look at these efficiencies, then partitioning efficiency is about 60%. Interception efficiency, and this is photosynthetically active radiation, is almost up to 90%. So what this tells us is there's not much room for improving these two numbers. And we're left with this one, the conversion efficiency. However, probably what we should compare those against is the theoretical maxima. So if we do that, then if we look before the Green Revolution, we can see that interception efficiency is around about 50%, partitioning efficiency below that. After the Green Revolution, in our major crops, rice, wheat, soy in particular, we see that the big improvement really is in harvest index or partitioning efficiency, where from something like rice we've gone from 30% to 60%, same in wheat, soybean not far off that either. And you know, if we think, well, if we're going to have some pod to hold our soybean, some stems to support it, we're not going to get a great deal further with that. Interception efficiency, speed at which the crop covers the ground, has also been improved by breeding as well. But what's changed very little is conversion efficiency. And what analyses we have on conversion efficiency suggest improvement there is largely increase in stomatal conductance, allowing more CO2 in, but actually lowering the efficiency with which the crop uses water. Now, of course, the major determinant of conversion efficiency is photosynthesis. So if we could improve this, could we actually get an improvement in yield? And you know, one of the questions we're often asked is, well, breeder selection and evolution surely have selected for photosynthesis already to be optimal. So did evolution leave a free lunch on the table? Well, I'd argue that selection is very much about survival and fecundity, and many of you are familiar with Arabidopsis, which is a very efficient winter annual in Europe in particular, and yet it's remarkably unproductive in terms of biomass production. Its photosynthetic rates are also pretty low. Another big change is that if we assume that our crop ancestors evolved over the last 25 million years, they evolved in a CO2 concentration of about 220 ppm. Today the atmosphere is almost double that. It's a limiting substrate for C3 photosynthesis. So actually in the last 50 years, half of the increase has just occurred in that time, which gives very few generations for adaptation to that change. Also, if we look at our crop ancestors, for example, this is one of the wheat ancestors. These are often plants growing in semi-arid, limiting conditions where most of the leaves are in full sunlight. Today we grow these in dense canopies, often leaf area indices of 6 or 7, where most of this is in shade. So at least half of our carbon is coming from photosynthesis in shade or shaded conditions. So the environment has changed a great deal, and I'd argue, and our modelling suggests that, in fact, often the situation is now suboptimal. Now, I come back to the question of, well, if we could improve photosynthesis, would we really get an increase in yield? And this is one of the free air concentration enrichment experiments. This is on our farm in Illinois. And so within this ring, the CO2 concentration from sowing the crop through to dry down of the crop is elevated to 600 parts per million to simulate the future atmosphere. This is a replicated block experiment. So what we find is when we first set this up in 2001, we were using in here what was then the most popular genotype used by farmers in our area. We got an increase in photosynthesis over the growing season of about 25%, and yield increased 15%. The reason why it didn't increase 25% was that there was some decrease in harvest index. Now, our breeder, Brian Deers, has come up with a very productive cultivar which is now being used, and Lisa Ainsworth tested that in this experiment 2013-2015, and in fact got the full 25% improvement and not a drop in harvest index. So there's evidence there that if under well-watered conditions we boost photosynthesis with elevated CO2, we get more yield. And similar experiments using the same system in China have shown this with rice now, and also experiments with wheat have shown this with wheat as well. So, okay, are there ways in which we might be able to do this without waiting for CO2 to rise and all the problems that come along with that? Well, photosynthesis is the best known of plant processes, I'd argue. There's been the subject of intensive biochemical, molecular, physiological research. We have high performance. It's also very conserved across C3 species. We also have high performance computing, which allows us now to simulate the whole process computationally. And crop transformation is becoming increasingly routine, even in the public domain. So one of the things that we've been working on for many years was to try and simulate the whole photosynthetic process, taking each of the discrete steps of photosynthesis, represented by a differential equation, and then integrate that system to start to predict how interventions would affect the outcome. Now, that, I make it sound an easy process, but actually Jingwan Zhu, who's my graduate student, worked over 4 years on this. And we had a lot of problems with the numerical integration. And we were discussing this one day at the computer center, and one of the mathematicians there said, "Oh, I have exactly the same problem with balancing rocket motors in my NASA project." And so, and this is how I solved it. So we used his routine, and luckily that actually solved our problem as well, and we were now able to get solutions. One of the things he did say, though, point out, we're often told plant biology isn't rocket science, but he actually said, but your problem's much more complex than mine, and I'd like to work with you on it because it's a much more interesting mathematical problem. So that's why we have a rocket scientist as our second author on this paper. So, but once we were able to do that, then we could start to apply optimization routines and start to make predictions of, well, where are the bottlenecks in this? Where might we be able to get efficiency improvements? But the problem, of course, when you move to a crop canopy is that conditions are changing all the time. Light changes every hour of the day, and as the crop grows, a leaf that started off in full sunlight is now going into deeper and deeper and more intermittent shade. And so you also have to take that aspect into account in making predictions. But we did that over a number of years, and we came up tongue-in-cheek with what increases you might be able to get. And S indicates this is something that you might be able to change on a relatively short time scale. So we're making these predictions, and fortunately for us, the Gates Foundation started to get interested in them, and they said, well, we're willing to put our money where your mouth is for a little while, and you show us this can actually be done. So we then had to really put a team together who were willing to go where no sensible plant scientist had been before, and that includes, so Chris Niogi from Berkeley, Susanna from here, TJ from CSRO, oh sorry, Don Ort from, from Illinois, Christine Rains from the University of Essex, Carl Benacki and Lisa Ainsworth from USDA ARS, and Martin Parry from the University of Lancaster. So our kind of mode of action was, we would make, of the billions of computational possibilities there, we would try and predict what might be the best targets. So really narrow down all the possibilities and make some predictions, and then transform those into tobacco. Now, you wouldn't think the Gates Foundation has any interest in tobacco, and they don't. The reason why we use tobacco is it's very quick to transform. It produces large numbers of seed, so we can quickly go from transformation to field trial in a way we couldn't do with soybean or cowpea or cassava, for example. But it does form a canopy, as does soybean and so on. So that sets it apart from Arabidopsis, for example, and it has high photosynthetic rates as well, so we're not improving on something with low rates already. So we would test this in controlled environments. If that looks good, then we move to replicated field trials, as you can see here. Now, if it passes that test, as some things have, then we go on to the crops the Gates Foundation care about, cowpea, where we're collaborating with TJ, and with rice, for example. We also have funding from FFAR, which commits us to also doing this in soybean. So through this modeling, we came up with, and discussions with our team, we came up with 7 possible pipelines. All of those go into 1 or 2 cultivars of tobacco, which we test at a common location. So we can then say, well, out of what we're testing, what looks most promising to move on to the target crops? So I'm just going to, sorry, I should say, we're just going to tell you about 2 of these: accelerating non-photochemical quenching relaxation, which is in my lab, and then lowering photorespiratory costs, which is in Don Ort's lab, and Don will talk about that aspect. So non-photochemical quenching, leaves of crop canopies, the upper leaves, are receiving, if they're in full sunlight, they're receiving far more light energy than they can actually use in photosynthesis. But the chlorophylls absorb those, go into an excited state, and if that energy is not dissipated, they can react with molecular oxygen to produce singlet oxygen and then a series of radicals which would literally bleach the leaf if those were not removed. And of course, evolution has come up with a way of dealing with that. Pretty well all plants do this. They induce a process which removes that excess energy as heat. So in full sunlight, a large proportion of the absorbed energy is dissipated as heat, and in proportion to that, there's a small amount of fluorescence, which is useful to us because that will tell us how much non-photochemical quenching is occurring. Now, if a leaf, and this really happens at the chloroplast level, if it goes into shade, then for several minutes, even though it could now use all of that energy in photosynthesis, it carries on dissipating a large proportion of as heat. And it takes many minutes before that process relaxes, and then it starts to use that in photosynthesis. So essentially what is happening is when the leaf goes into the shade, of course photosynthesis drops because there's less light, but it drops far further than it needs to, and it takes several minutes for that to recover. Now, of course, this will happen when clouds cross the sun, but actually far more important is what's going on in the canopy. As the sun moves across the sky, leaves above other leaves move, so you get this intermittent shading. And we simulated this quite a while ago, and what was happening, and then what the cost of this slow recovery would be. And we came up with a cost, depending on the plant, but a cost around about 20%. And at that time got talking to Chris and Iogi at Berkeley, who worked on the processes affecting this recovery in Arabidopsis. And so with our modeling, we'd come up with the fact that we needed to upregulate these two proteins. And from Chris's experience, he knew that upregulating PSBS would attenuate the effect of this. So zeaxanthin is closely involved in the dissipation of that excess energy. And so if you go into the shade, you need to remove the zeaxanthin. But you can't just upregulate this, because if you do that, you also make the plant vulnerable to photo damage. So we reasoned from the modeling that we had to upregulate both of these genes, even though this one might have sounded counterintuitive. Chris's experience added in this one. So we set about doing that, and two postdocs in my lab, Vanna Kromdijk and Kasia Glauacker, really led most of this work. And so we then put a construct that Chris had developed into tobacco, and these are three different events, and you can see this is the mRNA, so we'd got in these three events, those were expressing. This is the protein, and so we've then got, we could see that we've strongly upregulated ZEP and VDE, and also upregulated the PSBS. And when we analysed these plants, indeed, we were accelerating this recovery. And if we looked at photosynthesis efficiency under steady state, we hadn't affected that, but under fluctuating conditions, we had improved the efficiency considerably. So they looked good in controlled environments, so we moved to field trials, and in those field trials, we achieved about a 20% improvement. Now that is just in above ground biomass. We're not saying that if we now put that into cowpea, as TJ has now, that we'd necessarily get the same improvement in seed yield, but it's a strong indication we will get quite a significant improvement. And after a discussion with Science, who said, well, we don't normally publish things on field productivity of crops, they did actually then put it on the front cover, which was quite pleasing. One of course, the other things we were asking was, what do the individual genes do? And we got a surprise with the PSBS that, which we upregulated individually. We got a range of, we both downregulated it with RNAi, upregulated it with extra copies. And what we could do from that was produce a range of variation in QA, an important electron transport step, so we could change the redox state of that. And a surprising result came out was there was a very tight correlation with stomatal conductance. And this actually scaled with photosynthesis. And our modeling had said that actually we could reduce stomatal conductance a small amount without much impact on photosynthesis, but with an impact on water use. And so we, again, did field trials, and at the leaf level, we were able to get this effect, that we could get about a 15% improvement in leaf water use efficiency. Now, obviously, we haven't carried that through to lysimeter trials, but we have the ability to now do that. So at this point, I'll finish and pass over to Don to talk about what he's done on photorespiratory bypass. So, yeah.
Is that about right? Wow, big crowd. It's fun to be here, and as Steve said, I'm going to talk about one of the 9 or 8 pipelines, the photorespiratory bypass, and talk about lowering the cost of photorespiration. And maybe this is unnecessary for this crowd, but I thought I'd just introduce photorespiration a little bit. And so what we really want photosynthesis to do is when the CO2 comes in, Rubisco attaches it onto an RUBP, and we make 2,3 carbon sugars, phosphoglycerate. And of course, that's where the name of C3 photosynthesis comes from. But in reality, what happens is about 20% of the time, Rubisco grabs an oxygen molecule rather than a CO2 molecule. And that starts this process of photorespiration that can really be thought of as kind of a process of anti-photosynthesis. And so what happens then, it just makes one molecule of this phosphoglycerate, but it makes this molecule glycolate, and this is a 2-carbon molecule. And it has 2 things wrong with it. One, because it has 2 carbons, it can't be reincorporated back into the photosynthetic cycle. And then the second thing is that it's actually toxic to several of the enzymes in the chloroplast that carry out the photosynthetic carbon reduction cycle. And so plants had to deal with this once the atmosphere was oxygenated, and the way that they chose to deal with it was to recruit a lot of biochemistry that was going on and modify it in a way that it could recycle and detoxify this molecule glycolate. And plants figured out a very, very complex way to do it. And so glycolate is taken out of the chloroplast, it goes into the peroxisome, and from the peroxisome it goes into the mitochondria. And what it achieves is that it takes 2 molecules of glycolate, converts it back into 1 molecule of glycerate that can go back into the photosynthetic carbon reduction cycle, and evolves a molecule of CO2, and it also evolves ammonia. And so both of these have to be recaptured. And the cost of this, the energetic cost of this, is big. And so for every oxygenation reaction, it requires an additional 3.5 ATPs and 2 NADPH equivalents. And so we were curious of, how big is that from the standpoint of the total energetic flux of ATP and NADPH in the plant. And so we used a multi-layer canopy model that we parameterized with information from field studies done in Illinois on soybean. And so this is for a soybean canopy with a leaf area index of about 3, and so it's a full canopy. What you can see is this model predicts that during midday, about 40% of all the ATP used in the plant is used in photorespiration, and on the order of about 30% of the NADPH. And so this is not a side reaction by any means. This is a central reaction of central metabolism in plants, and is very costly. And so, what does that mean about yield? And so we went on and we used a process-based agri-ecosystem model, and again parameterized it with actual field data from soybean, and just asked the question, in the hypothetical case of no photorespiration versus the photorespiration that we get for the particular conditions in the growing areas of soybean in the United States, what is the yield drag? So what you can see here is this model predicts that in the Midwest, and Steve already told you that about 40% of the soybean grown in the world is grown in the U.S. Corn Belt, there's a yield loss of on the order of 35 to 40%. Furthermore, this model predicts as you go further south into the growing regions where it gets warmer and drier, so this process-based model then predicts that some of these yield losses could be as much as 50 to 60%. And so remember, this is the hypothetical case of no photorespiration versus the photorespiration that actually occurs. And so one approach to this would be to try to re-engineer Rubisco in a way that it does less of this oxygenation reaction. And certainly that's been the holy grail of bioengineering and photosynthesis. There's been a lot of effort at it. It hasn't been successful so far. Doesn't mean it won't be successful in the future. But the approach that we took was, rather than to try to do that, we were inspired by work that had been done earlier in Arabidopsis to try to install a more energy efficient way to metabolize glycolate and recycle it and detoxify it. And so we wanted to install these pathways into chloroplast, and we worked on 3 pathways. And so one of them is this pathway here. And so this is the glycolate oxidation pathway from E. coli. It involves 5 gene products from 5 different genes. And so we installed that into tobacco chloroplasts. And then we used 2 completely synthetic pathways. One in which we used the glycolate oxidase from higher plants, which normally functions in the peroxisome, but we moved it into the chloroplast. It uses oxygen as an acceptor, and so it makes hydrogen peroxide. So we had to deal with the hydrogen peroxide, so we put a whole lot of catalase in there as well. And then finally we put in malate synthase to make, so the glycolate and acetyl-CoA make malate, and then that's decarboxylated. And then the other synthetic pathway, we used glycolate dehydrogenase from Chlamydomonas. The advantage of that is it doesn't use oxygen for electron acceptor. It's not abundantly clear what it does use. It was thought for a while that it used NADH, but it probably donates the electron right into the electron transport chain of chloroplast at the level of quinone. But at any rate, it doesn't generate hydrogen peroxide, so we didn't have to put in this massive amount of catalase. And so I'm going to tell you mostly about this pathway 3, where we've used the glycolate oxidase, or the glycolate dehydrogenase from Chlamydomonas. And I'll talk a little bit about this pathway, because these were the 2 most successful in our work. The other thing that we wanted to do is we wanted to force as much of this glycolate flux as possible through what we think were going to be the more efficient pathways and away from the native pathway. So the way we thought to do this was to downregulate the exporter of glycolate out of the chloroplast. And that had been identified a few years before we started this work by Andreas Weber's lab, and it's a exchanger of glycolate and glycerate, and it's known as PLGG1. And so we knocked that down with RNAi. And so we did these experiments both with and without these RNA modules to knock down that transport activity. The other thing that really made this work possible was the ability to make big complexes in labs that weren't historically molecular biology labs. And so we used Golden Gate, and we were able to put together these rather large constructs as large as 30 kb and get the entire pathway in a single transformation. The other thing nice about Golden Gate, it's very modular. And so, for instance, if we have 5 genes, we can put on 5 different promoters, 5 different terminators. But then, since we don't know a priori what kind of expression we're going to get, what kind of activity is optimal, within the Golden Gate, we're able to move these. And so, for instance, here we've moved this gene 5 to the front, and so each one of these now has a different promoter. And so all in all, we put together 24 different constructs with and without the RNA targeting for PLGG1. And we did at least 10 independent transformants of each one. And so we ended up with a whole lot of plants. And we needed to be able to deal with these and get them down to a few number of plants that we thought were promising candidates. And so we used a technique that was developed here by Murray Badger. We call this the Badger Box 2. And what we wanted to do is, our notion was that if these pathways are active, they should be photoprotective. And so we wanted to put these plants under very high photorespiratory stress, and the notion being that an active bypass pathway or alternative pathway would rescue them under this stress. And so here's an empty vector plant, and so we're measuring this parameter FV prime over FM prime, and in a healthy situation you're going to get a value between 0.7 and 0.8. And so here's our empty vectors at ambient CO2, and you can see they're pretty healthy. Then we put them in the Badger box for 24 hours at 1200 microeinsteins and a CO2 concentration that's below about 50 parts per million. Remember, atmospheric's about 400 parts per million. So very strong photorespiratory stress. You can see these plants really had bottomed out in terms of their photosynthetic efficiency. But here, for instance, is just one of the transformation events for this AP3 pathway. And now you can see again in the ambient situation, but now in the low situation, we have a significant amount of photoprotection. And so we were able to screen this way and get down from tens of thousands of plants to a manageable number of plants to work with. And this just shows some of the data, some of the range of data that we got out of this kind of screen. And so each one of these is an independent transformation event. And so this is this AP3 line with and without the RNAi module for the transporter. So you can see that in some cases we're seeing an improvement, we're seeing photoprotection, other cases we don't, and in some cases there's an indication that this module that forces more of this total glycolate flux through the alternative pathway might show an advantage. And so this is the way that we selected the plants that we took forward. And so I'm going to talk most, as I said, about the AP3 pathway. And these are the data that shows that our genes are expressed. And so we're looking here at the relative expression level. Here is the Chlamydomonas GDH, malate synthase. And you can see also that our PLGG1 RNAi really knocks down this transporter about 80%. Here we're looking at the localization of the proteins. These we expect to be in the chloroplast. So here's what we see in the whole leaf. When we get into the chloroplast, you can see that we're seeing an expression of the glycolate dehydrogenase in the chloroplast, a lot of malate synthase in the chloroplast. And then these are our markers for the chloroplast and markers for the cytoplasm. We were a little surprised here that we weren't seeing more of the dehydrogenase, but there was information in the literature that some of it bound to the chloroplast membrane, which would make sense if it's using the electron transport chain as its electron sink. And in fact, we found quite a bit of it associated with the membrane. And so these proteins are there. They seem to be functional based on our screens for photoprotection. The other thing that we expected, that if we were putting a lot of this glycolate through this more energetically efficient bypass pathway, we should be able to see it in the efficiency of photosynthesis. And so we looked at the quantum yield of CO2 fixation, which you can see here is the quantum yield of the wild type or the empty vector under either normal atmospheric CO2 concentrations or very high photorespiratory stress. What you can see is in many cases we're seeing statistically significant increases in the efficiency of photosynthesis as judged from the quantum yield of photosynthesis measured at low light intensities. We also wanted to look at some of the metabolite profiles, and so we looked at some of the metabolite profiles for molecules that are signature molecules for the photorespiratory pathway, the native photorespiratory pathway, as well as for our introduced pathway. And so what you can see is that levels of glycolate and glyoxylate go up with the AP3 pathway installed. The glycine to serine ratio increases. And then pyruvate, which is a major product of our alternative pathway, also goes up. We did these, but I don't think that they're particularly informative because this is just a snapshot in time and we're looking at just pool sizes. And so really what we want to go on to do, and we started a collaboration with Doug Allen at the Danforth Center to do more flux analysis, because what we really want to know is the fluxes of these compounds, not just what their concentration is at a point in time. But this is at least consistent with what's seen in other photorespiratory mutants. And so at the end of the day, we wanted to know what this does to the whole plant, and so we started out with greenhouse studies. In the greenhouse studies, we see that all of these three pathways drive a significant increase in, well, they drive an apparent increase in plant dry matter biomass. And so this is the E. coli pathway, and again, with and without RNAi. I bring your attention to the fact that in the case of this AP1, the E. coli pathway, there seems to be, if you knock out the transporter, it seems to drive down the stimulation of yield. This AP2 pathway, this is the one that had the plant glycolate dehydrogenase installed in the chloroplast, where we're making a lot of hydrogen peroxide. This never performed very well, and as I said earlier, the best performer was the AP3 pathway. And so in this case, in the greenhouse studies, you can see that we're seeing increases in dry matter biomass of 20 to 25%. And again, this just shows some of the variation that you see among the independent transformants. These are the AP3 transformants. And so in some cases, we're seeing significant increases, others not. Sometimes we see differentiation, or we see lower yields. And in this case, we're beginning to see that the PLGG1 RNAi in concert with this AP3 pathway seems to drive higher increases in biomass, consistent with the notion the more glycolate that we run through this pathway, the more benefit that there is. And so at the end of the day, what Steve said, we really want to get these out in the field and see if they function in the field under conditions of the very high photorespiratory stress that you can get in the field and the variation in conditions that you get in the field. These were irrigated experiments, and so in this case, they weren't complicated by whether or not there was water deficits. And so this is just a picture of planning out the replicated field study. Here's the biomass information from the 1960 field trial for the E. coli pathway. And what you can see here again is we see, if we just look at total biomass, we're looking at 3 different independent events. In 2 of the independent events, we're seeing significant increases in the total biomass, not in this 3rd one. But again, we're seeing in the case of when we put in the RNAi module to knock down the glycolate transporter, we're actually seeing a depression of what's going on, of the increase in yield. And what we think, and we have some evidence for it, and we'll be following up on this, is we don't believe that the pathway, the E. coli pathway, that as we've installed it, has enough flux capacity to deal with the entire glycolate flux that's produced by photorespiration under the field conditions. And so then when we knock out the transporter, the glycolate concentration would go up in the chloroplast and become inhibitory. And so, that's our reasoning right now. In the case of AP3, again, we're seeing some quite large increases in total biomass of 20, in this case 25%. But in this case, when we put in the PLGG1, we don't see a depression, but we don't necessarily see a statistically significant difference between the 2. And so what we wanted to do in the 2017 field trial was to increase our statistical power. And so we focused just down on AP3. We increased our replication number up to 6 to increase statistical power, to see if we could get results that could differentiate between when we knocked out PLGG1 and when we didn't. And so this is what we saw in the 2017 field trial. And so now you can see this biomass difference compared to wild type. We're seeing in the absence of the RNA module that knocks out the transporter about a 30% increase. And when we install this module, it increases to about 40%. Once again, we see the same increase in the quantum yield that we saw in the greenhouse studies. We see an increase in the daily integral of photosynthesis, and starch concentrations go up. And so what's next? And so one of the things that's next is we want to move these into crops that we care about and the Gates Foundation care about. And so our target crops right now are soybean, cowpea, cassava, and potato. I already mentioned that we've engaged with Doug Allen at the Danforth Center to do metabolic flux analysis to really validate the biochemistry that's going on. I mean, one of the things we know is we're producing a lot of malate, we're producing a lot of pyruvate in the chloroplast. So we know the chloroplast has exporters for those 2 molecules. And so some amount of it is going out, and some of it could be going into the TCA cycle, some into glyconeogenesis. And so we'd really like to know the biochemistry that's really going on, and that's the reason to do that. I told you about PLGG1 in terms of being a glycolate exporter, but we discovered a second one, and the second one is BAS6. And so we also want to knock that down separately and in combination with PLGG1. And we have those plants now at the T0 state. And then finally, we also want to start combining some of this. Some of the modeling that Steve has been doing suggests that some of these different pipelines, when combined together, should be synergistic. And so right now, Steve and I are collaborating to put the AP3 bypass along with the non-photochemical quenching together. And so that's work that's underway. And so, as I said, this work that's underway, these are cassava plants that have Steve's construct in it. And so we've gotten these from the Gates Gas Project. This work was done in Willie Gruesom's lab. Here are some of the cowpeas that have Steve's construct in it that T.J. Higgins has done. And here are rice plants that have that we've transformed at Illinois that also have the construct in it. And so, of course, the notion is that by collaborating with TJ, it possibly gives us a route to get these plants, once we get them into cowpea and some of these other plants, into field trials in Africa. And so that's an aspiration of something we'd like to do in this 5-year study. Whether we'll get there, we'll just have to see. And finally, I just wanted to point out people that did the work. And so Paul South led most of the molecular biology. Another postdoc in the lab, Amanda Cavanaugh, did a lot of the field physiology. Berkeley Walker, who's now as a faculty position at Michigan State, did most of the modeling. And then 2 other shout-outs. Dave Dragg is our field manager, and so most of the field studies wouldn't have been possible without him. And someone not in this picture is Claire Benjamin, and so she's our communications coordinator and is probably the reason that people have heard about this work. And so let me end there. Thank you.